Hello, everyone. Welcome to our Translational Neuroimaging Educational Program. I'm the moderator for this event today. My name is Yang Ge, and I work at the University of British Columbia. Before introducing our guest speaker, please allow me for like 2 minutes to have a quick introduction of this educational program. And this education program is nested within the lifespan working group of the Enigma Consortium, which spans over 45 countries and involves over 4,000 researchers worldwide. This program is an inclusive one that welcomes researchers at all levels of neuroimaging skills and from diverse training backgrounds. The director of this program, Dr. Xiaoxiao Li from Mount Sinai, New York City, and myself, and we do have an international advisory committee. Please consider joining our mailing list if you wish to receive notification for our updates about our program. We have a YouTube channel where you can find the recordings from previous events. I'm also recording today's event. So if you are interested, please keep an eye on our YouTube channel, and we will be uploading very soon. Just a heads up, we will be only sharing the presentation by our guest speaker, but not the Q&A part. So please feel free to raise any questions for our speaker after their presentation. We are both on Twitter and Blue Sky, and if you are on either platform, we would love you to follow us there. And if you wish to share your work through this platform, please get in touch with us. We welcome any type of collaborations. And our next event will be on March 11th. Well, Young He from USC will join us to share their latest work on a 3D brain geometry toolbox. And today, we're very excited to welcome Yanting Yang. Yanting is a PhD student in the Department of Electrical and Computer Engineering at University of British Columbia, advised by Dr. Xiaoxiao Li. His research aims to improve the fairness and efficiency of AI models, translating advanced machine learning algorithms into real-world healthcare applications. His work primarily involves developing novel deep learning architectures to analyze complex neuroimaging with a specific focus on the functional MRI. Today, Yanting will introduce his work on deep learning for personalized and interpretable fMRI. And we're really looking forward to learning about your new work, Yanting. Now the microphone is yours. Yeah. Stop sharing.
Great, thanks for your introduction. And let me share my screen. Okay, let me know if you can see the screen.
Yes.
Okay, great. Good morning, everyone. My name is Yanting Yang. I'm a PhD student in the Department of Electrical and Computer Engineering at the University of British Columbia. I work in the Trusted and Efficient AI Lab, supervised by Professor Xiaoxiao Li. Today, I'm excited to present two of our recent works that address key challenges in applying deep learning to functional MRI analysis. The first work is called FunFormer, which discovers personalized brain functional networks, and the second is NeuroLIP, which achieves interpretable and fair cross-modality alignment between fMRI data and phenotypic text. Here is the agenda for today. I will start with an introduction covering the background and motivation for this line of research. Then I will present the FunFormer, our brain functional network transformer for discovering personalized functional networks. After that, I will present NeuroLIP, our cross-modality alignment framework that is both interpretable and fair. And I will conclude with a live demonstration of both systems, followed by a summary and a conclusion. And the talk shall take about 35 to 40 minutes, leaving time for the demonstrations. And I will first go into the introduction. So let me first start with some background. The functional MRI or fMRI capture the brain's distributed activity through blood oxygen level dependent signals. By computing the statistical dependency between the time series of different brain regions, we can construct functional connectivity matrices that encode the brain's network architecture. Deep learning has shown tremendous potential for extracting clinically relevant biomarkers from this connectivity data. However, there are two fundamental open challenges. First is personalization. Most existing methods rely on a fixed group level template that obscure the rich variability between individuals. The question is how to capture both population-level consistency and individual variability simultaneously. And second is the fairness. Models trained on biased datasets may learn to exploit sensitive demographic attributes like age or sex as proxies for disease prediction, rather than learning disease-specific neural signatures. Today's talk addresses both of these challenges through our two proposed frameworks. So before diving into the methods, let me briefly review the concept of functional connectivity. The standard pipeline begins by parcellating the brain into a set of regions of interest, or ROIs, using a brain atlas. Suppose we use the CC200 atlas, which defines 200 spatially coherent regions. For each ROI, we extract the average both time series and then compute the Pearson correlation between every pair of ROIs. This gives us a symmetric 200 by 200 functional connectivity matrix, where each entry quantifies the strength of co-activation between two brain regions. And these FC matrices are valuable because they capture the brain's intrinsic network organization. They have been used to study the disrupted circuits in conditions like autism spectrum disorder, the ADHD, and Alzheimer's disease. And importantly, the FC patterns are also sufficiently unique to serve as neural fingerprint that can identify individuals. Our two papers build directly on this foundation. The first FunFormer uses FC matrices to discover personalized functional networks, and the second NeuroLIP aligns them with phenotypic text for interpretable and fair classification. And let me visualize the two challenges side by side in detail. So on the left, we see the personalized challenge. The existing group-based method assumes uniform functional networks to all individuals, capturing the general scaffold, but washing out the specific variations that are actually informative for diagnosis. On the other hand, individual-based methods like subject-level clustering capture the individual differences but often produce biologically unrealistic mappings. On the right, we see the fairness challenge. The existing cross-modality alignment methods project an entire fMRI image and its phenotypic text into a single global vector. This approach discards spatial details and, more critically, entangles disease-related features with sensitive demographic attributes. This means the model might learn to predict autism partly from age or Alzheimer's partly from sex, rather than learning disease-specific neural signatures. Our two works address these challenges respectively. The FunFormer tackles the personalized challenges, and the NeuroLIP tackles the fairness challenge. So I will first go through the first proposed work, the FunFormer. And to introduce FunFormer, our brain functional network transformer. The goal of FunFormer is to simultaneously uncover both stable group-level functional network scaffolds and meaningful individual specific deviations from fMRI functional connectivity data. And the key insight behind FunFormer is that the brain's functional organization is hierarchically structured. There is a stable group-level scaffold that is broadly conserved across individuals, but the most predictive and clinically relevant information resides in the subtle, individualized ways in which each person's functional network organization deviates from this shared scaffold. And our hypothesis is that these structured, personalized deviations are not noise, but carry meaningful biological signal. We validated the FunFormer on 3 multi-site datasets, the ABIDE dataset with over 1,100 individuals for autism classification, and the ADHD 200 dataset with 767 individuals for ADHD classification, and the ABCD dataset with nearly 1,750 participants for cognition score prediction. And here is the architecture of FunFormer. The input is the subject-specific functional connectivity matrix. Each row of this matrix represents the connectivity profile of a single brain region, and it is projected through an MLP encoder into an embedding space. These embedded vectors are our node token X. And the key innovation is the introduction of the learnable prompt tokens P. These are k-trainable vectors that serve as dynamic centroids for the functional networks we aim to discover. And unlike fixed clustering approaches, these prompts are learned end-to-end through backpropagation and can adapt to capture the optimal network structure for downstream prediction. And the node tokens X and the prompt tokens P are concatenated and fed into a modified transformer encoder layer. We apply a mask to prevent prompt tokens from attending to each other, which encourages them to develop into distinct cluster centroids. And after the transformer layer, the deep embedded clustering module softly assigns each node token X to the prompt token P based on their similarity, producing a soft assignment matrix A. This assignment matrix A defines each individual's functional network organization, and the merged representation Z are then passed to a readout layer for the final prediction. Let me explain the prompt-based clustering mechanism in more detail. So first, we introduce k-learnable prompt tokens that function as latent network attractor in the embedding space. And these prompts are randomly initialized and updated through backpropagation during training. And the prompts P are concatenated with the node token X and jointly processed by a modified multi-head attention layer in the transformer model. And a crucial design choice is that we masked the prompt-to-prompt attention. The prompt tokens cannot attend to each other. This encourages each prompt to develop into a distinct cluster centroid by focusing its attention solely on the node tokens. And after the attention layer, the deep embedding clustering module computes the similarity between each node token X and each prompt token P, producing a soft assignment matrix A. This is a probability distribution indicating how strongly each brain region affiliates with each functional network. There are two key properties. First, the same brain region can affiliate with different networks depending on the individual, reflecting the brain's conditional network membership. And second, the group level scaffold is not imposed as prior knowledge, but emerges organically from the aggregation of these individual solutions. And we also add the orthogonality and the entropy losses to ensure the prompts remain distinct and the assignment are well calibrated. And this figure visualizes the functional network discovered by the FunFormer on the ABCD dataset. The row labeled group shows the consensus network organization aggregated across all test subjects. You can see that the group level network are bilaterally symmetric and aligned with established canonical functional systems like default mode networks and the frontal parietal network. And the row labeled subject 1 and subject 2 shows the individual level network organization. While the overall structure is preserved, you can see clear subject-specific deviations. Each individual has a unique pattern and how their brain regions are assigned to functional networks. Furthermore, the network organization is dynamic, looking at the same subject across the rest and the MID task condition. You can see the distinct network configurations, particularly in frontal regions. This demonstrates that the FunFormer captures both the stable scaffold and the context-dependent personalized reorganization of functional networks. And to quantify the cohesiveness of the discovered network, we compute a group-level co-association matrix. This matrix stores the probability that any two brain regions are assigned to the same functional network across all subjects in the test set. And the chart diagrams has visualized the top 1% of these co-association probabilities. And you can see that the links are overwhelmingly concentrated within clusters rather than between clusters. The intra-network connections are markedly more probable than the inter-network connections. This strong modularity confirms that the FunFormer robustly groups brain regions into cohesive functional communities, reinforce the biological plausibility of the discovered networks. And a critical question is whether the individualized deviations captured by FunFormer are stable and distinctive enough to serve as neural fingerprint. We tested this by attempting to identify subjects across different scanning sessions using their personalized network patterns. The panel A shows identification accuracy across different session pairs, the rest to task and the task to task. Across all conditions, the individualized functional network representations enables reliable subject identifications well above the chance level. Notably, the task-based fMRI condition consistently yield higher identification accuracy than the resting state. And the panel B shows how many brain nodes contributed to identification under each condition. The task-based fMRI involved a larger number of subject-specific nodes, suggesting that cognitive engagement amplifies the expression of individualized network organization. And interestingly, one network identified by our LLM-assisted interpretation as frontal parietal network consistently yield the highest identification accuracy across all session pairs, indicating that individual differences are concentrated in higher order cognitive networks. And in terms of predictive performance, we compare the FunFormer against several deep learning method baselines, including BrainNet-CNN, the FBNet-G, and BrainNet-TF, and ConBrain-TF. And we also compare it to the two-stage hybrid approaches that use the standard clustering algorithms for feature extraction, followed by a prediction hard. This table summarizes the result across all three datasets. The FunFormer achieves the strongest overall performance across ASD classification, ADHD classification, and cognitive score prediction. The key takeaways is that the personalized network representations are not merely descriptive. They provide network-level features that are necessary for capturing the clinically and cognitively relevant variabilities in the data. Models that preserve individualized network organization consistently achieve stronger performance than baselines that either operate directly on connectivity matrices or rely on two-stage clustering pipelines. To establish the biological validity of the discovered networks, we performed the term-based meta-analysis using the NeuroSync database. The NeuroSync maps brain coordinates to cognitive terms based on co-activation across over 14,000 neuroimaging studies. And the figure shows the top 10 most frequent cognitive terms associated with the predictive networks for each downstream task. For ASD classification, the most predictive networks were associated with sensor processing and executive control, which is consistent with documented ASD symptoms. And for ADHD, the network were linked to attention, motor, and memory-related functions, aligned with the core deficits in ADHD. And for cognition score prediction in the ABCD dataset, the network were enriched for working memory, cognitive control, and language, consistent with the cognitive domains assessed. And these results confirm the functional networks discovered by FunFormer are not arbitrary partitions, but reflect biologically meaningful functional systems. And to understand which specific networks derive the predictions, we use the integrated gradient and axiomatic model interpretability method. This figure shows the predictive power of each network for ASD classification, ADHD classification, and cognitive score prediction. And a finding is that the prediction score depends disproportionately on a subset of functional networks. For ASD, the networks 7 and 8 made the most significant positive contributions, and our meta-analysis identifies these networks as being strongly associated with executive function, consistent with the well-documented executive function challenges in autism. And for ADHD, the network 6 was markedly more important than all others, aligned with the cognitive terms memory, a core cognitive domain implied in ADHD. And we also perform a case stratified deviation analysis, which reveals that the correctly classified cases exhibit systematically lower deviations from the group consensus compared to the misclassified cases. This suggests that adherence to canonical network structure facilitates the accurate classification. And this figure shows the within-network functional connectivity differences between disease and the control group. In a BITE dataset in panel A, the ASD subject exhibited significantly reduced within-network connectivity in most networks, consistent with the well-documented hypoconnectivity profile in autism. And in contrast, the ADHD subjects in panel B shows increased within-network connectivity, reflecting a different pattern of altered intrinsic connectivity dynamics. And crucially, in panel C, it shows the clinical relevance of the individual network deviations. We compute the deviation score as the adjusted mutual information between each patient's network assignment and the group level consensus. And for a BITE dataset in the upper figure, the network deviation was significantly correlated with the ADOS total score, meaning greater departure from the canonical network organizations was linked to increased ASD symptom severity. And similarly, for ADHD 200 dataset, the network deviation was associated with higher ADHD index scores. This result demonstrates that the individualized network deviations captured by FunFormer are not only predictive of diagnostic group membership, but also scale with clinical symptom severity. And our ablation study confirms the importance of key architecture components. Removing the learnable prompt tokens results in a marked reduction in performance, confirming they are essential for capturing individual network organization. The optimal number of cluster was k equals 8, which was significantly better than other choices. This suggests a natural granularity of network population for capturing diagnostic signals. And for the test-retest reliability, we analyzed the ABCD dataset across 4 fMRI conditions between the old trials and the even trials. The overlap matrices show strong diagonal patterns, indicating high stability and reproducibility of the identified functional communities across independent measurements. This confirms that the personalized networks are not driven by noise, but reflect robust features of functional brain organization. So let me summarize the key takeaways from FunFormer. First, the FunFormer reveals that the brain's functional organization is hierarchically structured with a stable group level scaffold supporting systematic individual deviations. Second, these individualized deviations serve as reliable neural fingerprints that are stable and reproducible across cognitive states. And third, the personalized Third, the personalized networks are clinically relevant, improving prediction of neurodevelopmental disorders and correlating with symptom severity. And fourth, the discovered networks are biologically valid, aligning with canonical functional systems confirmed by the NeuroSIMS meta-analysis. And the fifth, the learnable prompt tokens provided a methodological advance, resolving the tension between group consistency and individual specificity in functional brain network analysis. And next, I will introduce the second proposed framework, the NeuroLIP. So now let me turn to the second work, NeuroLIP. The motivation comes from a fundamental limitation of existing cross-modality contrastive learning methods like the SIGLIP and the CLIP when applied to neuroimaging. As shown in the figure panel A, the existing method projects an entire fMRI connectivity matrix and its corresponding phenotypic text into a single global vectors, which are then aligned through a contrastive learning loss. This approach has two major problems. First, it obscures the fine-grained relationships between specific brain regions and textual descriptors. We cannot tell which brain regions are activated by which phenotypic trait. And second, it entangles the disease-related features with sensitive demographic attributes. The model cannot distinguish whether it is predicting autism based on neural patterns or based on age. And the NeuroLIP addresses both problems through the token-level alignment. Instead of compressing everything into a single vector, we preserve the brain region level tokens and align them with individual text tokens. This design enables both fine-grained interpretability through attention maps and intrinsic fairness by separating diagnostic features from demographic confounders. And here is the overall architecture of NeuroLIP. The framework employs two parallel encoders, a vision transformer for the fMRI connectivity data, and the biomed BERT for the phenotypic text. The fMRI image encoder produces per patch embeddings, where each patch corresponds to one brain region. And the text encoder produces per token embeddings for each phenotypic attributes. And the key innovation is the cross-modality alignment via localized tokens, or the CALT strategy. Instead of aligning global vectors only, we introduce the text token conditions the attention, or the TTCA mechanism. The TTCA uses the text tokens embeddings as queries and the brain region embeddings as keys and values in a cross-attention layer. This produces text-conditioned image features. For each text token, like autism or male, we get a corresponding image feature that represents which brain regions are most responsive to that particular attribute. We then apply three types of losses: a global contrastive loss for overall alignment, a localized CALT loss for fine-grained attributes level alignment, and an attention distance loss for debiasing. Let me explain each of these in the next slide. So first, let me explain the text token condition attention mechanism in more detail. So the TTCA is essentially a cross-attention layer where the text token embeddings serve as queries and the brain region embedding serves as keys and values. And for each text token, whether it represents a disease label like autism, or a demographic attribute like male, or an age group like child, the TTCA computes the attention weights over all n brain regions. And these weights indicates which brain regions are most responsive to that particular text tokens. This produces text conditioned image features that have the same dimensionality as the text token embeddings, enabling the direct token level comparison. And the key advantage is that a given brain region may receive high attention when conditioned on the disease token, like autism, but low attention when conditioned on the sex token, like male. This is what enables the explicit separation of disease-related and demographic associations. And because the attention weights are directly interpretable, we can visualize exactly which brain regions the model considers relevant for each attribute. And the CALT loss is our localized contrastive learning objective. At the global level, we apply a standard sigmoid contrastive loss to align the overall image and text representations. But the key innovation is the localized CALT loss. For each attribute in the phenotypic text, like the disease label, the age group, and the sex, we compute a separate contrastive loss at token level. The CALT loss aligns the text conditioned image features for a given attribute with the corresponding text token embeddings. The positive pairs are matched image text samples, and the negative pairs are unmatched samples. And an important design choice is that we use separate learnable temperature parameters for each attribute. This allows the model to adaptively calibrate the sharpness of similarity distribution for different phenotypic features. For instance, the model might learn a sharper temperature for the disease label and a softer one for the less discriminative attributes. And now let me explain the biasing mechanism. We propose two complementary approaches that work together. The first is the negative gradient technique. Instead of encouraging the model to align image features with sensitive attribute features, we actively discourage such alignment by reversing the sign of the CALT loss for the sensitive attributes. This reverses the gradient direction during back propagation, driving the model away from learning correlations between brain representations and the sensitive demographic information like sex or age. And the second is the attention distance loss. We maximize the L2 distance between the attention weight vector for the disease token and the sensitive attribute token. This explicitly encouraged the model to attend to different brain regions when processing disease versus demographic information, ensuring these two types of information are treated as distinct. And the overall loss function combines the global contrastive loss, the localized CALT loss summed over normal attributes, the negative CALT loss for the sensitive attributes, and the negative attention distance loss. The negative sign before the lambda 2 and the lambda 3 implement the debiasing. Minimizing the negative of these losses is equivalent to maximizing them. And let me show you the quantitative results. This table summarizes the classification and fairness performance across all three datasets. I'm highlighting the AUC for classification accuracy, the DPD for demographic parity difference, which measure fairness, and the ES-AUC, which balance both. The NeuroLIP achieves the highest AUC on all three datasets, and at the same time, it achieved the lowest demographic parity difference on all three datasets. Compared to the CLIP model, the NeuroLIP reduced the DPD by 58.6% on the Abide dataset, 69.4% on the ADHD 200 dataset, and 70.7% on the Acne dataset. This is a critical result that it demonstrates that the fine-grained alignment and the fairness can be jointly optimized without sacrificing classification accuracy. In fact, NeuroLIP improves both accuracy and fairness simultaneously because separating disease-relevant features from demographic confounders actually helps the model learn more disease-specific neurofilament signatures. And our ablation studies systematically evaluates the contributions of each debiasing components. The tables result on the byte dataset with 4 configurations: the no debiasing, the attention loss only, the negative gradient only, and both combined. Each component individually reduced the demographic bias, adding the attention loss alone, reducing the DPD from 11.6 to 10.5. Adding the negative gradient alone reduced the DPD from 11.6 to the 9.4, but their combination yields the best overall performance with the lowest DPD of 7.9 and the lowest DODDS of 16.7 and the highest AUC of 72.4. And this combined effect arises because the two mechanisms are complementary. The attention loss ensures that the model learns distinct attention patterns for disease versus sensitive attributes at the representation level. The negative gradient prevents the encoding of sensitive information during the actual alignment process. Together, they enable NeuroLIP to achieve superior fairness without sacrificing classification accuracy. And one of the most powerful features of NeuroLIP is the interpretability provided by the TDCA attention maps. This figure shows the brain surface projection of the attention maps generated by the conditioning on the disease tokens versus control tokens across all 3 datasets. For the byte, the ASD conditioned attention map highlights regions spanning the default model network, the frontal parietal network, the attention networks. Regions that support language processing, working memory, and attention and episodic memory, and all of which are disrupted in autism. For the ADHD 200 dataset, the ADHD condition maps densely sample the default mode network, the frontal parietal network, both dorsal and the virtual attention networks. The prioritized functions, attention, executive control, working memory, and motor regulations map directly onto the core ADHD deficits. And for the Anthony dataset, the Alzheimer's disease condition maps prominently feature the DMN limbic system, particularly the hippocampus and the hippocampal gyros, along with the temporal and parietal regions. These are the cardinal regions affected in the Alzheimer's disease. The attention maps clearly show the disease-specific patterns that are anatomically plausible. And to validate the clinical plausibility of our attention map, we combined spatial correlation between NeuroLIP's attention maps and the NeuroSIM's topic-based meta-analytic maps. The radar plot shows the top 10 NeuroSIM terms with the highest correlation values for each dataset. And for ASD, the strongest correlations are with speech and the language-related process, along with face recognition, which is consistent with the well-documented language and face processing deficits in autism. And for ADHD, the dominant correlations are with actions and motor execution domains, as well as attention and working memory, directly mirroring the dual deficit model of ADHD. For Alzheimer's disease, the top correlations span memory retrieval, language comprehension, and social cognition, which represent the cardinal cognitive deficit in AD, and the strongest agreement between our model attention pattern and established meta-analytical findings across all 3 disorders provide compelling evidence that the NeuroLIP captures neuroanatomically meaningful patterns rather than spurious correlations. This is critical for building trust in AI systems for clinical neuroimaging. And let me summarize the key takeaways for NeuroLIP. First, the CLT the CLT strategy with token level alignment preserved the region brain specificity that global alignment method described. And second, the TTC attention map provides interpretability by localizing disease-associated brain regions. And these patterns are validated against NeuroSIMS meta-analytic data. And third, the two debiasing mechanism, the negative gradient and the attention distance loss, substantially reduced demographic parity difference by up to 70.7%. And fourth, the NeuroLIP achieves state-of-the-art classification performance on all three datasets. And fifth, perhaps the most important insight, the fine-grained alignment and fairness are not in conflict, but actually reinforce each other. By separating disease features from demographic confounders, the model learns robust disease-specific neurosignatures. And next is the demonstration and conclusions. So let me move to the in the next few minutes. Let me move to the live demonstration portion. We have built the interactive gradient web applications for both the FunFormer and the NeuroLIP demos. They are all publicly available as Docker images on the GitHub container registry. And the FunFormer demos supports two tasks: the disease classification for autism and ADHD, and the cognition score prediction for REST, MID, and back-end SST. User should upload the ROI time series files in the CC200 parcellations with 200 regions. And the demo runs five-fold ensemble inference and provide the group level visualization, including a co-association matrix, the chord diagram, the consensus brain parcellation, and a 3D interactive viewer for the brain. And at the subjective level, the user can inspect the individual predictions confidence score, the ARI score, and download all results as Nifty file. And for the NeuroLIP demo, it can perform disease classification across three disorders: the autism, the ADHD, and Alzheimer's disease. In addition to the ROI time series, users should provide phenotypic metadata in the CSV format, specifying the diagnosis sex and the age group. The model can leverage the biomarkers for the text encoding and the TTCM mechanism to produce the separate attention maps conditioned on the diagnosed sex and the age tokens. These maps are rendered as interactive 3D brain surfaces projections and can be downloaded as Nifty files for further analysis. And both demos are containerized and can be launched with a single Docker command. And let me switch to the live demonstration. First, if you look into this Git repository, it contains all the instructions on how to start the demo. You only need to implement these two lines of code to start the demo, as long as you have the Docker installed in your system. And also, GPU is not needed for this demo. And if you are working remotely in a server, you need to do the port forwarding via the SSH using this command. And after you execute these two commands, you can directly use the web browser to open the applications. And here is the example of the applications. So here in the input panel, you can select the task and the disease type. For the disease classification, you can select autism or ADHD. For the cognition score prediction, you can select 4 kinds of fMRI tasks. And here, for example, we select disease classification and for autism, and we drop in some example files for this demo, like we drop for 1, 2, 3, 4, 5. We drop 5 files, and it will show a file summaries and a time point distribution for you to check if the loading is a success. And then you can click the run button, and it will run the group analysis over all these 5 input ROI time series. And due to the lightweight parameter of our model, the execution will be very fast. And here is the a summarization of the summarization table, which includes the prediction, the confidence, and the ARI scores. And you can download these tables. And we show 3 different plots. The first is a co-association matrix. The second is the chart diagram. And the third is the consensus network postulations, which is interactive, and you can see it at different coordinates. And the colors are corresponds the colors of each in each figures corresponds to each other. So you can directly see that the network A corresponds to these regions, which contains these brain regions. And yes, you can also find it in the interactive viewer. And also you can download the group postulations in the NII format. And in addition to that, we provide the network ROI assignment. For each network, we provide the ROI index for that. We provide the ROI index within each network, and we also provide the ROI names in the AAL format, so that you can do further analysis based on this table. And also you can download this table here. And the next is the subject level analysis. In this drop-down menu, you can select either a single subject, and it will report the specific scores for that subject. For example, the prediction is 0, which means it's healthy controls, and the confidence score is 1, and the ARI score is 0.12. And we show here the group versus the subject level network confusion matrix to see how the subject level network deviated from the group level networks. And in the right figure, which also shows an interactive viewer for you to see the networks in the brain. And you can also download this in NLI format. And also we provide the network ROI assignments similar to the previous one, and you can also download this here. And that's for the FunFormer demo. And I will show you how to run the code in the second demo. So here, in the first demo, I did not show you how to run the code. And in this demo, I will show you. So first, I will just queue the last demo. And it will close the service. I will clear the terminal, and I will run the NeuroLIP demo, which is exactly the command that is listed in the GitHub. And I will just run this command. And shortly, it will show that you can open the web browser, and the demo will work. And next, let me switch to the web browser. Here, I refresh the pages, so now it will be the NeuroLIP demo. And in this demo, you not only need to upload the ROI time series, you also need to upload the phenotype informations. So here I already have the phenotype information here ready. So I will copy and paste it here. And to be noticed that the phenotype information must contain the following columns: the name, the diagnosis, the sex, and the age groups. And all the diagnosis, sex, and age groups are binary labels. And the name shall be exactly the name of your uploaded time series. And to be noticed that diagnosis is not actually needed. So if you did not have it, it's totally fine. I can just delete it in some regions, in some rows, but the sex and the age groups is needed. So I will upload some files using the NeuroLIP test set. For example, I will also upload 5 files here. It will also shows the file summary, like here. And you should note that it's totally fine if you did not provide diagnosis, because it will not be used in the following visualization. And we click the Run button. This model may take a longer time than the FunFormer because its parameter is larger than the FunFormer. So here it generates the summarization report in this table with the predictions and the confidence score. And for each, and also in this drop-down menu, you can click either individual subject, and it will generate the visualizations of attention map that is conditioned on the diagnosis, conditions on the sex, and conditions on the age groups. And you can download all these fields as Nifty files. Okay. Now let me go back to the presentation. Yes, here is the demonstration of our apps. And yes, here's the FunFormer demo with the subject level analysis, the NeuroLIP demo with the subject level visualization. And here, let me bring everything together with a summary. On the left, the FunFormer captures the individualized functional network signatures through learnable prompt tokens that act as dynamic network attractors. As a result, the long-standing trade-off between group-level consistency and individual specificity in functional brain network analysis. The personalized deviations is discovered correlated with clinical severity and enable robust neural fingerprinting across cognitive states. On the right, the NeuroLIP achieves interpretable and fair cross-modality alignment between fMRI data and phenotypic text. STTCA mechanism enables token-level attribution between brain regions and phenotypic traits. The debiasing mechanisms reduce demographic disparity by up to 70.7%, while simultaneously improving classification accuracy. And the attention maps are validated by NeuroSync meta-analytical data. And the unified themes across both worksets, the deep learning can move beyond pure prediction accuracy to provide personalized, interpretable, and fair analysis of brain function. These are essential properties for translating AI into clinical neuroimaging practice. And thank you very much for your attention. And I hope this talk can give you a sense of how deep learning can be used to provide personalized and interpretable and fair analysis of functional brain image data. And I will be happy to take any questions or discussions regarding this work. And you can also go to these GitHub reports to check the demos.